think we're good to go. Wait like one or 2 minutes, just in case. I'm just gonna copy the link. Do you know how to access like the Facebook, or sorry, the YouTube channel for HMG360? And do you know how to access the YouTube HMG360 page? Joanna, are you carrying around a camera?
No.
No? What is that? Looks like a camera strap.
Wait, where? No, this is my.
Oh. I'm just nosy. Apparently that really worked. I got the link. Thanks, Jeff. Let's see how it goes. Actually, I'm going to try to use this time to maybe focus your studies just a little bit so that you're not studying the wrong sorts of things. And we're actually live, is that right, Jeff? Yeah. Okay, I think I have to get to the other. And how do I get to the other screen? Okay, Alt-Tab. It's this one that I'm presenting on, right? Okay. So hopefully this all records and we're doing an audio backup and we'll see how things go. In the worst case scenario, I'll also post the notes for you separately. So this review is more or less designed just to give any of you who are sort of doing last minute preparations for the final exam a little bit more focus. And if you've already started, maybe to make sure that you're studying the right sorts of things for your final exam as well. So just a couple of really brief comments about this. The multiple choice section is not trivial. It's challenging. So I'll just mention that from the get-go, that it's both comprehensive and fairly detailed in the types of questions that I'm going to be asking you. So the material, again, I haven't had many questions about this, just one, but it's everything that was not covered on the midterm exam. It is not a cumulative final exam. It's 2 hours, but it's fairly long in terms of writing, mainly because you're going to be writing more than you really need to. There are space restrictions, but I'm going to ask you to think a little bit, spend 1 or 2 minutes thinking about how you're going to answer your question before you actually begin writing it. So you'll have 20 multiple choice test questions worth 1 mark each, and then 4 short answers worth 10 marks each. So your final exam will be out of 60. For each of the short answers, you'll get a block of space that you'll be writing in. So again, just a couple of things. The multiple choice itself is fairly detailed. They'll ask you specific things from the bullet points on your slides. And the short answers are more of a big picture. Have you actually understood why you use a certain technique or why you might do a certain type of analysis when you go back and do the short answer questions? So they're very different in terms of the philosophy. Multiple choice for detail, and then short answers to make sure that you understood and can tell me how you might do a certain type of experiments. In many ways, just like the short answer questions that you have on discussion board. You have any questions about the format or anything else before I go on? And I'm assuming there's a long delay online or on Facebook. And is it actually streaming, Jeff? Do you know?
Let's see.
Hoping that it's streaming?
It's good, yeah, it's going.
Okay, so just a couple of general comments before I go through lecture by lecture the types of things that you need to do. So the multiple choice test questions that I like having on the final are integrative. So it's not just, which of the following would you find in a Zika virus, right? So that's an example of a question that is not integrative. You might get some information that leads you to suspect that you're looking at Zika virus infected neurons or progenitor cells, and then you'll have like things that you might have to solve about that. But the multiple choice are integrative. Generally speaking, there's only like 2 questions that are really like the first type, where it's straightforward, where you just drop down a list and pick things out. So other things that you will need to know for your multiple choice. For the multiple choice test questions that you get on your final exam, many of them are related to the polymorphisms. So which of the following polymorphisms are going to be involved in a scenario that you might be getting? So you need to be clear about the different polymorphisms, whether it's related to schizophrenia. So for example, you might get information. I'm not going to be asking you like, tell me about the epidemiology of schizophrenia. You might get information like, um, an individual has a condition where on an MRI they have less activity in the frontal lobes, they have issues with hallucinations, they have issues with delusions, etc., etc. It occurred when they were in their late teens. Which of the following polymorphisms might you expect to observe in this individual? Okay, or which proteins might you expect? So it's not about schizophrenia itself. It's about understanding that we're talking about schizophrenia, then being able to pick up these the precise sorts of polymorphisms that might be associated with it. Okay, so it's more of that type of question. I'm not going to ask you like which of the following statistics about schizophrenia are incorrect. So know about the different polymorphisms, enzyme systems that we discussed in class related to things like schizophrenia, prion disorders, and there are a number of prion disorders that we'll go through. There are a few questions that relate to specific methodology that will be useful for you in your multiple choice as well. So for example, a good type of question might be, an individual has identified a macromolecule associated with a neurodegenerative condition or a condition, and they treat that with PNGAs. And you might not know what PNGAs is, but if you do remember what PNGAs is used for, then that multiple choice test question becomes easy. Do you guys remember PNGAs?
Deglycosylates.
So it deglycosylates, right? So when we want to remove sugar moieties or sugar groups from our proteins, it tells us a little bit about what system we're likely working with. And in this particular case, it might be something related to a prion disorder. So again, knowing specific methodology wherever possible will help you a lot in terms of your multiple choice test questions. When we're talking about the different types of tests, whether it's a diagnostic test or a different type of analysis, make sure that you know the basis, what the fundamental technique is, what is sort of being examined when you go through for the multiple choice. And as I said, the multiple choice will be the more challenging in terms of you being able to apply what we've learned in terms of the way in which you're being asked different questions, okay? Anne, do you have a question?
Still don't really understand what you mean by polymorphism. Like, are you looking for a specific like SNPs or?
It could be a SNP, but a polymorphism might also be something related to, for example, in schizophrenia, we talk about different polymorphisms associated with the C4 gene. So there are multiple isoforms of that. Okay. Anything else that you want to chat about before we go on to the next slide? And again, I'm hoping this is recording well for you, and the link will be made available for you at the end, and we'll have all of the recording audio as well as the slides available for you at the end. So just out of curiosity, before I go on, how many of you have gone through all your notes at least once? Okay, not bad. If you had to write the exam tomorrow, what would your mark be? Pass? Okay. All right. Well, we'll hope that this helps you study a little bit more logically as we go on. So these are lists of things that I want you to study for the multiple choice test questions. And I want you to be able to go back over your notes if you haven't already. These are specific things that I want you to study for the multiple choice. I want you to know about the different types of sequencing methodologies. So you should go back, figure out like what sorts of things are associated with different types of sequencing. We mentioned, if you have gone through that lecture, we talk about quality assurance. Do you guys remember that lecture? Like, how do we make sure that the sequencing is good? And there are about 3 or 4 slides. You should know those slides. Those will be important for you in your multiple choice. They may also help you in your short answers. As I mentioned to you before, we talk a lot about different SNPs or polymorphisms. Some of these are related to prions. So we talk about specific prions, alpha-synuclein variations, for example, the one variation that we talk about a lot for the AST or alpha-synuclein, schizophrenia variations. C. elegans, you should go back over the genes that we talk about for C. elegans. We talk about C. elegans more than once in the last few lectures, okay? So we talk about it when we talk about sleep, and then we talk about it for RNA inheritance. And in both systems, we talk about specific genes, and I'll get back to that at the end, okay? So what I'm trying to do is get you to start thinking about the multiple choice being very, very specific. And I'll be asking you specific things related to C. elegans genes. It's not just something that we've glossed over. We talk about it, and it's related to a methodology. For example, in C. elegans, can you tell me what a somatic gene that is normally associated in the studies that we looked at the very last lecture with C. elegans? Can you name me any? But by the 28th, you should be able to tell me a couple of genes that are somatic or germline that are associated with C. elegans. And that's what I mean by being specific and being able to apply it to these different scenarios related to C. elegans genes as well as the RNA processing. Anything I can clear up for you on this slide before we go on? Is there anything from the online group, if anyone is watching online, or from the Facebook group, if the thing is up there on Facebook?
May I ask you a question? It's not about the exam though.
It's not about the exam?
Yeah.
Okay.
Are we gonna get our marks back about that 4% participation assigned questions?
Basically, all I'm looking for is whether you've completed it or not, and I'll be posting those pretty soon.
Okay.
Um, just one thing. There is a calculation question. You don't actually have to calculate it, but you should know how to calculate something. It's kind of, again, the title of this was ominous music here. You might be thinking, where did we have calculations? And it's related to sequencing. I'm not going to. We had Oriana, we had calculations in sequencing. There's lots. There are lots of things that you should know how to be able to calculate different types of things. And I'll just give you a heads up because I don't want it to be something that is lost in translation. You don't actually have to do the calculation. There's no need for you to bring a calculator, but you should be able to know how to calculate different things. What a value actually means or how it might be calculated. There's also a bonus question at the end of the exam. There's no penalty for trying it. There are 4 different sort of slots that you can just write a short answer in, and that's your bonus question. And you can do it, and then you'll get a 1% bonus if you complete it. Any questions on this before we go on? My hope would be that you do everything first before you try the bonus question, though. Like, don't waste time on it. It's not, unless you know it right off the top, it's not worth actually sitting there doing. The short answers are more important that you fill those out. So for your short answers, just a couple of things I'm looking for when you're doing your short answer questions. Give specific examples wherever you can in your answers. So if there is, don't say promoter, be specific. Tell me the type of promoter that you're going to be using. If you're going to be developing an animal model system, which is very common in the types of short answers I give you, specify the type of animal model, why you're using a specific mouse strain, for example, why you're using C. elegans. Again, if you're using C. elegans, why you might use a specific genetic marker for C. elegans. tell me a little bit about the rationale. It doesn't have to be a long thing, just a few sentences. You know, this is a good mouse model because this hemizygous mouse actually doesn't exhibit neurodegeneration, but it's more vulnerable for neurodegeneration. So you have both limited space and limited time, so be very wise in what you start writing. Don't go back and try to erase everything and then go back. The other thing, the opposite way to think about this, don't leave anything blank. Okay, so I can't give you marks if that whole sheet is blank, even if it's a couple of points that you start off with and you're not 100% sure. If you're blanking out, just jot stuff down. Make sure that it's part of the actual space that you're provided. Don't jot things down in the margins, like put it into the space and then come back to the question later to think about it. But don't leave anything blank. That's the worst thing that you can do, is get 0 out of 10. It happens a lot in the final exam, probably more frequently than you would think, but you should try not to leave anything blank. I'm happy to try to give you part marks if it makes sense to the question that's being asked. You may get the animal model wrong, but your reasoning, your rationale might be right, and you'll get a couple of marks for that. Any questions about the short answer questions? Again, we have 4 of them. We'll go through this in some more detail as we go on. Nothing? Multiple choice, okay. Short answer, okay. Scared, not so scared. Really scared? Really scared, okay. So I'm going to go through the lectures section by section. So for the schizophrenia lectures, the very first ones that you're responsible for, February 9th is the lecture date that I have in my notes. Don't bother memorizing any of the epidemiology. Again, I'm not going to be asking you, tell me about schizophrenia. It affects 1 in 100 individuals, affects males more than females. There's a prodromal period, all of that. You should know that. It's part of your it might be part of the scenario that you're getting, but I'm not going to be asking you to tell me that on your short answers, I guarantee you. And in terms of asking you specific multiple choice related to this, it's not going to be like, which of the following numbers about schizophrenia is wrong. Don't memorize the epidemiology. Understand what describes schizophrenia, but don't bother with that. The dopamine system, I'm going to be very honest, slide 11, slide 12, I don't have questions related to the neuroanatomy of the dopaminergic system. Okay, don't bother memorizing it. Something that you might have gotten before. It was there to give you some context. Don't bother memorizing it. You might want to go back and review a little bit about the autoreceptor, the D1 autoreceptor, sorry, the D2 autoreceptor, and then go back and think a little bit about the dopaminergic system as a whole, but don't bother memorizing those slides. Really, there's nothing on it. And I don't want you to be wasting your time. You have a few days now before you write your final exam. My job is to get you ready for your final exam. Okay, so when we talk about specific small details, these are not the small details that you'll get on your multiple choice. Is that okay for everyone? Okay, so things that you should know. Remember, we talked about things related to understanding the big picture for the short answer questions. Multiple choice is about fine details and understanding and being able to tell me a little bit about how much you remember about specific methodologies and other things. You should be able, for the big picture for schizophrenia, tell me a little bit about the glutamate model hypothesis in schizophrenia. You should be able to tell me a couple of drugs that we talked about. Do you remember any of the drugs related to schizophrenia? Because you might get that on your short answer question. Any of the drugs? Ketamine is something related to the glutamate model hypothesis, and it's been used to induce symptoms that look like schizophrenia, but any other drugs that are used to treat schizophrenia that we talked about? I told you not to remember the whole list of things, but there was one sort of classic drug. Yeah. So haloperidol was one of those different types of drugs. Might be something to go back and revisit a little bit, especially with the dopamine hypothesis of schizophrenia. So this lecture was kind of an overview to get us into genome-wide association studies. And at the end, there were a number of different criteria in this lecture associated with GWAS. And you should go back. Don't leave it, because it's something that you need to know. about the criteria that we use to make sure that a GWAS study is a good GWAS study, okay? So again, there's not as much on this lecture on schizophrenia as you think. It was sort of an introductory lecture to get you into schizophrenia and then into GWAS. Don't go through the epidemiology, don't go through the neuroanatomy, talk about haloperidol, where it's sort of binding, how it tells us about what dopamine might be affecting, and then this glutamate model in schizophrenia. Anything else before we move on to the next slide? Or anything that I can clear up for you on this slide? Okay. So the next lecture that followed after this one was the GWAS lectures, starting on March 1st. You should know the strengths as well as the weaknesses, both for multiple choice and for short answer. You should know the strengths and weaknesses of using GWAS studies. We can't just randomly use GWAS studies. It works really well. In some instances, there's some reasons for why we want to use them, but there are also other reasons why we don't use them in certain types of studies. You don't have to memorize. There's like a list of, I think, two to three slides that go through the differences and the advantages in case control studies, cohort GWAS studies, and trio studies. You don't have to memorize that. You don't have to memorize the minutiae. I would just be familiar with what a cohort GWAS study is, what a case control GWAS study is, and then what a trio study is. When you take your mom, dad, and then the proband, or the affected individual, that's sort of the types of things that I'd want you to know. I mentioned to you earlier, when we talk about GWAS, you should know the quality indicators. There are a number of them. It's not just one. We go through a number of different quality indicators that tells us if a GWAS study is being performed correctly, if it's a good GWAS study, if the genetic sequencing is done right. All of those things you should know. So quality indicators, yes, means that you should go over those slides. Do you guys know the slides that I'm talking about? Kind of, you can sort of visualize it.
Can I ask a question while we're
Yeah,
on the topic?
sure.
So on the first slide where you have the SNPs, you said something about you want 80 to 90% success rate.
Yeah.
And then the next slide, you said the student call rate should be above 95. I'm wondering, is that the same thing or is it?
It's slightly different. It's the way that the sequencing is done. We didn't go into a lot of detail about call rates and everything else. And again, you don't have to worry so much about the overall numbers that you're reporting back. I would say that on average, the higher the number, obviously, the better it is for any of these GWAS studies. And nowadays, 95% as a call rate is pretty routine.
But we don't need to know the detail of like what it is and how to.
Not for call rates, because I didn't go into call rates a lot. So basically, when you go through and you do your sequencing, you're able to make the genetic identity of the particular fragment of DNA that you're looking at. You're able to identify it fairly accurately. If there is that same stretch of DNA that you can't tie back to a gene, then that doesn't fall into that call rate. So generally, we're able to identify with the same fragment or the same piece of DNA. So if we've broken it up this way and we've identified this little stretch here, that would give us sort of a call rate of like 100%. And if we had one that was just outside and we're not 100% sure, then that would give us sort of 5 out of 6, 83%. Okay? And you're doing this for multiple fragments. Sorry, Kay, did you have a question?
I just wanted to ask about the cohort because I was confused with the cohort study. Is it about taking like a group of people you don't know if they'll have the disease or not, and then you genotype them? Do you categorize them based on their genotype, or are you categorizing them? Because you also said you observed to see if they.
Yeah, you can do it either way based on their genotype or on their phenotype and see what sorts of things come out based on what happens after you look at the two different cohorts of individuals, whether it's on phenotype or on genotype. The case control is the one that we're more used to, where we actually look at these individuals have a disease, these individuals do not. Let's see what the differences are. But the other one, you just want to see, like in this whole population here that we have in this classroom or at the University of Toronto, let's just see what differences come up, and then let's see how we can segregate those different individuals. So it's a slightly different approach to different types of things. And again, this goes back in many instances when we're looking at different types of studies. Some are hypothesis driven, some are not hypothesis driven. Are GWAS studies generally hypothesis driven or not? No, so they're not hypothesis driven. And the way that we set these different types of studies up also determines how we approach a particular hypothesis. Okay, so different things like this. So these definitions, if you know what they are, or you can sort of work your way through them, these will go a long way to helping you on your final exam. As I mentioned, there are certain things I'd like you to make sure that you know. We talked about the R2 value or R squared value, and then the linkage disequilibrium. These will come in handy for your final exam, and just knowing the definitions will go a long way to helping you on your final exam multiple choice. And then these C4 gene variations that we get to right at the very end, those will be things that you probably want to go back and review a little bit.
Does the big R squared and the little R squared mean the same?
No, it's the same thing. I just use them sort of interchangeably. Well, I use them interchangeably.
Association?
Okay. So then the next part of the lecture gets into schizophrenia mechanisms. So this is the March 3rd lecture. So this was the Tuesday. The following Thursday, we did the March 3rd lecture. What I'm going to ask you to do is know the major details of the paper. Be able to summarize what did they find. That's really it. A summary of what they found, the different techniques that they were using throughout the particular paper. So usually I take you through the different types of stains that were used, like PSD95, the Hooks stain or DAPI stain, staining for the C4, and seeing if it localizes pre, post-synaptically, or in both places. This is an example of a figure you don't have to memorize. So you don't have to memorize this. I'm not going to be asking you this. If you were, though, to go back and pick one protein that you've seen in this list of things, which one would be the most important one for you to know?
C3 or C4?
Right, so we go through in this particular paper, C4, you should go back and look at the C3. That's a big part of this paper too. And then there are other things that they mention at the very beginning of their paper, and I talk about it at the very beginning of this lecture, related to interact, what sort of has been known before about the C4 gene. There are other things that have been shown to interact, not just C3, other things as well. Okay?
I heard ask for the lecture before that.
Yep.
I never really knew the relationship between CSMD1 and the C4 gene. Because I remember we had confusion on like one of your slides would say it was like the CSMD1 would be regulated by the C4 gene, and then we asked you.
I think the more important thing is that there is sort of an interaction between the two. Not, you don't need to know the directionality of it. So which one influences the other one more, but that there are things that influence C4, if that makes any sense. Okay. Okay. And again, make sure that, as I mentioned at the very outset, some of you came in a little bit later. You need to know how to apply different techniques. So, you know, why do we stain for things like PSD95? It's a postsynaptic density protein. If it co-localizes with PSD95, that usually means that it's found postsynaptically. We might look at things like V-glut1 and 2. And why would we look at V-glut1 and 2? So it's a presynaptic marker, and if it's found co-localizing with the presynaptic marker, especially in glutamatergic neurons, it tells us a lot there as well. Okay, so the next lectures that we get into are the ones on prions. So do you think you have to know all of this? Yes, no? Should you memorize this? Should you memorize all this stuff here? Yes, no, maybe?
Not all.
Not all? Then how much of it?
Specific promoters and the most important proteins in that gene transfer powder.
Okay.
If you want to use it like as you mentioned in the short answer question, you want to apply stuff. So maybe it will be helpful for that question
Okay.
to design something.
Okay. So the consensus, do we need to memorize this or not? Yeah, Oriana?
To know that there's membrane associated.
Um, sure, I guess so. Um, I'll just be really straightforward. I don't like these types of questions in the sense that it's not something I spend an exhaustive amount of time on. I mentioned that there are a number of different regions associated with both the amyloid precursor protein. It was to set up that maybe they're not so different, and I didn't go into in a lot of details. So don't spend time memorizing like all of these different types of things. However, having said that, when we talk about human prion proteins, we actually mention specific single nucleotide polymorphisms that are associated with disease. And I highlighted them here to begin with, and then we talk about them. They're in your bullet points on your notes later on. So that is stuff that you should definitely make sure that you know, all right? So don't bother memorizing this diagram because I'm not going to be asking you to tell me, what are the most where do we get different kinds of variability in the N-terminal region? Which types do we see the most often? If it was a biochemistry class, I probably would, but not in neurogenomics. So again, in the March 8th lecture, no need to memorize everything. However, when you go back over your notes, you should know that there are some things that we identify as being sporadic. Right now, we don't have a good sort of genetic cause for it. So we don't know why it happens, but we don't think it's genetic. And there are other misfolded prion-related disorders that we suspect are very strongly genetic in origin. And you should know the differences between them and which ones. For example, Kuru is not a genetic prion disorder, but other things like Jakob Creutzfeldt might be. And again, I mentioned this at the very outset, so you should know, for example, about different types of techniques that we look at when we look at prions in particular. And in this particular case, we went through and we actually talked about this in class, about the role and the use of things like proteinase K, and whether or not we have a proteinase K resistant core, then that's a good sign that we have a misfolded and aberrantly misfolded prion-like protein. So that might be something that you see both on your short answer as well as in your multiple choice, because that's a method-related technique, and it's also something that might be useful when you're going back in talking about prion disorders, okay? Okay. All right, so let's go through the rest of the March 8th, and I'm hoping that all the nomenclature for the dates is correct. If I'm mistaken, you'll have to correct me a little bit later on, but when I made the slides, I kind of based it more or less on the dates that were set on your Blackboard page. So you should know what Lewy bodies are. You should know how to detect Lewy bodies. It's part of the notes that you have. Do you guys remember what MSA is? If you guys have been reviewing MSA? Yep. So multiple system atrophy, there's some clinical features associated with the disorder. Do you guys remember any of those? Yeah.
Orthostatic tension, autonomic.
Okay, so there's a whole bunch. Yeah. Okay, so good. I'm glad that people have been looking at this. So here's another example of a really busy slide that we took a look at. So do you have to memorize all of these? Yes, no, not sure. Yes, no, maybe. Chloe is saying no. Kay is kind of looking like, okay, fair enough. So you don't have to know inheritance patterns, whether it's autosomal dominant or recessive. I would just know like alpha-synuclein. Don't have to know chromosome location. Again, if this was a full-blown third-year genetics class, I would ask you to know the chromosomal location because they're important in the third-year genomics class. But in this particular context, it wasn't. Because what we did was we started to go in and we talked about prion proteins, and then we talked about maybe some of these other proteins that are associated with neurodegeneration, like alpha-synuclein. If it misfolds, maybe we could see it acting like a prion in a particular mouse model. So the alpha-synuclein is important. The locus, chromosome location, inheritance pattern, not so important. Again, this is also part of your notes. You don't need to go through, and I'm not going to be asking you about this. There is one thing to keep in mind, though, because it is genetics or genomics. I'll interchange, for example, alpha-synuclein with the SNCA gene. You should know that alpha-synuclein is derived from SNCA, and then we talk about one particular SNP. Do you guys remember what that was? Yep. So the A53T, if you see that, then yes, you should know that we're talking about alpha-synuclein or something that's related to the end product of SNCA, and all of those should be like things that you equate with each other. Okay, so if you see SNCA, it's most likely we're talking about alpha-synuclein. If you see things related to a polymorphism associated with diseases like Parkinson's, then we're talking about the A53T. And we haven't, and you don't have to memorize all these other ones because we didn't really talk about them. Does that clear things up a little bit about what you need to memorize, not memorize? No? Okay. So again, knowing how to detect Lewy bodies. We also talk about these mice. Do you remember the transgenic M53s? What were they? What might be their advantage in using them? So we had transgenic M53s, plus minuses, plus pluses, minus minuses. What are the advantages to using them? Yeah.
Like the plus they would develop the disease sporadically, but the plus minus would not unless they were inoculated with the infected protein.
Right, so the hemizygous mice don't have any degeneration normally occurring until they have an insult added to them. And that's one of the reasons why we use them when we're looking at prion-like disorders, which is what was sort of being done here. They took some of the proteins from MSA patients. They were looking to see if there was any effect on these mice with them. And you should go back, review these mice, because as I mentioned to you before, these mice models are really important. Yeah, Anne?
Where is M53 in the notes? I only see M83.
Well, it could be 83. I don't know. I typed it. I don't know. 83. I promise on the final exam, it says 83. If it says 83 in your notes, it says 83 there. I was just typing it really fast. Is that why everyone was kind of like, did we talk about this? Okay. My apologies. So this should be up here, the 83 instead of 53. Okay, so at the very end of the March 8th lecture, because we're then going into FFI, I talked very briefly about molecules that regulate sleep and structures in C. elegans that have been implicated with different types of behaviors. And it might sound like it's a throwaway at the very end of the lecture, but this is where I usually take multiple choice test questions. So it's worth going back and taking a look at the end part of the March 8th lecture related to molecules that regulate sleep. There are a number of them that have been implicated. People were looking for them. And then we talk about some other structures in C. elegans at the very end of the lecture. And then we segued into another prion disorder, FFI. Okay, I think I saw your hand up.
Yeah, I was wondering, I noticed in that prion lecture, you gave like a little introduction about like prions, and then you were talking about the hypothesis of like from the gut to the brain. I was wondering if we had to know about that.
No. No. If you take LMP 410, you might hear a little bit more about that in your fourth year. Anything else I can clear up for you? I'm happy to answer those types of questions. I don't want to be disingenuous. No one has a lot of time to mess around. You guys already have cognitive overload where you're feeling like you're memorizing an entire phone book anyways. So let's make the phone book a little bit more relevant for you. Anything else I can answer for you related to the types of questions that Kay just asked? I'm happy to answer them. Don't want you wasting your time. Okay. Um, we get into the next lecture, which I think was originally the March, it said March 14th, but it's really by date March 15th, I believe. Um, there are a number of different mouse models that we talk about when we get and we segue into fatal familial insomnia or FFI. There are mouse models that you should go back and make sure that you understand how they might be useful in this particular study. And there were a number of studies that were going on. There were also specific SNPs that we talk about related to fatal familial insomnia, and in this lecture, you should make sure that you go back over that as well. As I mentioned at the very outset, when we talk about multiple choice test questions, these are the types of things that you'll get lots of, specific SNPs. You might get methodologies, and again, this gets into methodologies, and specific results from the paper, which is related to this autophagosomes. Yep. Chloe, did you have a question?
No, I think I figured it
Okay.
out.
So what we did was we started with the orexin system or the hypocretin system. They're interchangeable. They're really the same thing. Two different groups came out at the same time. They identified this new peptide, new gene, and it turns out they actually had identified at the same exact same molecule. So some people call it orexin A, orexin B, and some people call it hypocretin 1, hypocretin 2. They're really the same thing. You should know why these molecules are important in sleep. There were specific HLA markers that were associated with these different isoforms or different genes that were associated with damaging these systems. You should make sure that you know those, which is the third point that's highlighted for you here at the bottom. It's not quite working its way down. So slow. There you go. All right. And you should know how to make sense of this. In this particular paper, we also go back over a couple of methodologies. And again, the thing I don't want this to be is really too stressful for you. So I do talk about specific methods in the lecture, and I want you to be able to tell me a little bit about whether or not you remember what context these were used in. In this case, we already talked about PNGAs, which is used to deglycosylate proteins, and you should go back over this as well. You should know some of the results about the autophagosomes and autophagolysosomes. And then the last thing, in terms of big picture, because I told you that there are two different strategies. One is I'm going to be testing you for lots of detail on the multiple choice, which is only 20 marks. And then I'm going to be asking you to tell me about how you understand these big picture things. And one of the big picture things was we have these new models and these new ideas about how other molecules, not just prions, but maybe other molecules like alpha-synuclein or other molecules that have been implicated in neurodegeneration, how they might look like prions, how they might cause other normal proteins to aberrantly misfold if they're injected into the brain. How do we test for infectivity? And if you can answer that, and it's also one of your practice short answer questions, then you'll go a long way to doing well on the short answer question. And I'll just leave it at that. You can think a little bit about that, how you might answer it, how you might actually demonstrate that you understand the rationale for what you're doing, the different types of mice that you might use or might not use, and then go on from there. But it is something to think a little bit about for your final exam. So the RNA sequencing lecture, we sort of spread it out over two different lectures that week. So the March 18th to the 22nd. This is one of the two lectures. If you haven't already, these lectures are ones that you should spend time on. Okay, so this one and then the one slide coming up after this. So this is one, if you haven't done any studying or you want to go back and really re-review things that are relevant for your final exam, this is the lecture that you should start with. Okay, so go spend some time on this. There's a lot of coverage on this. There's lots of material from this lecture on your final exam. So it starts off with stuff that gives us a little bit of background. Do you guys remember that interesting diagram that kind of looks like a wedge that looks like this, where we have differences in genetic variability, and we have SNPs all the way over here on the right, and then we have large chromosomal translocations and other things all the way down here. Big pieces of DNA moving, small single base pairs over here. Do you guys remember that? Do you guys remember that there are different techniques that actually allow you to more rapidly and more efficiently see things that are occurring down here versus over here? So karyotyping and other things might be really useful down here, but karyotyping won't detect a single nucleotide polymorphism. Do you guys remember that slide? It's worthwhile going back over that slide, at least for the multiple choice test questions, to make sure that you are okay with it. Okay, and if you understood all of that and you can apply all of that, then it'll be an easy question for you. So that's the second point here related to optimal techniques. So then we get into other techniques, including RNA sequencing. So when do you use it? Where do you use it? And why might it be useful for detecting what happens after Zika virus infections? And those are questions that hopefully will guide you in terms of how you're studying for this section. There's no need to memorize the tables related to the different viruses. I think there's a little green table with different DNA viruses, RNA viruses, I think, if I'm not mistaken, in this section. You don't have to memorize that. I'm not going to be asking you questions related to this. To be honest, not on here as a bullet point, but do you guys remember that life cycle of the virus as well? So how it attaches to the cell, the host cell attachment factors, how it enters the cell, all that, don't need to know it. Okay, so it might be useful for you on your final exam short answer question if you relate things back to it, but you're not going to have to memorize anything specifically from that. We talk about diagnostic tests. Do you guys remember any diagnostic tests related to this lecture? What is this condition that is associated with Zika? Chloe? So microcephaly diagnostic tests that we talked about? Do you guys remember any of those? Yep.
There was like the standard deviation, 2 standard deviation after birth, 24
Okay,
hours.
sure.
The other one was taking like placenta fluid and detecting certain proteins. I don't remember them specifically, but that's probably the thing of the test.
Yep, so detecting different types of proteins can tell us a lot about things like spina bifida. They can tell us about neural tube closure. If we're detecting excess acetylcholinesterase, for example, it tells us a lot. And those diagnostic tests related to the nervous system are ones that you should go back and make sure that you study for your final exam. Yep.
There were also other sequencing tests, like techniques that we talked about in class, like
Yep.
next-gen sequencing,
Yep.
Sanger sequencing,
Yep.
PCR, all
Yep.
that stuff.
Yep.
That will be also needed to know information.
Yep. Not going to ask you to tell me how it's done. It's not going to be a short answer question. If you're
Okay.
so if you're thinking short answer question, no. Should I have told you that? Too late. Okay. But yeah, you should be familiar with the different types of sequencing. But I'm not going to be asking you like, how do you do Sanger sequencing? What's the difference between Sanger and next gen sequencing? What's whole exome sequencing? No. The last one, the March 29th lecture, is really the other lecture that you want to spend time on and go over it carefully. Can you figure out why? Well, obviously because it's on your final exam, but can you figure out like why I would tell you that? Yeah.
Well, it was like a big deal since they were using a different technique to identify neurons. It was like new because we always
Yep.
just identify neurons based on physiology mostly, and
Yep. So all of those things. Now we can do it physiologically, electrophysiologically. We can do it anatomically by putting in, or histologically by putting in a marker, and now we can do it genetically. So all great things. But if you're thinking a little bit about this, when I'm telling you this is a big lecture to go through and think about, this is a big lecture to go through and think about, you're probably going to see lots of it on your final exam, which means you're probably going to see it both on your multiple choice and on your short answers. So you need to think about that a little bit. How might this be asked as a short answer question, or what sorts of things might be asked on a short answer question? I don't want it to be something that you feel is an unfair test, and it's not a trivial test by any means, and I'm not giving you all the details explicitly about what you should be doing, but this gives you some things to think about and prepare mentally for your final exam. Also associated with this paper, you should know the methodology associated with it. Like, how do you actually look at the histology of a cell that you're recording from? What do you inject? Or what sorts of things do you do? I don't need you to memorize all those little graphs, but you should know like what CCK is, what the other types of interneuron is, all the major bullet points, in other words, from the results. Those are things that you should go back and make sure that you're comfortable with. So you don't have to memorize. And this is what the 100% comprehensive. You don't have to memorize every last detail, but you should know like how the techniques were done. You should know what the major results were from this. And then you might want to start thinking about, if it is a short answer question, how you might rethink it and how you might redo or revisit things if you were asked to go back and do similar types of things. Okay? So it is a really important lecture that you spend some time going back over. Any other questions about this? I think we're sort of nearing the end. So the last one is on RNA inheritance, and I tried to do this one a little bit differently. So from this lecture on April the 5th, as posted for you on Blackboard, you should know slides 8 through 11. Okay, so go and review those. The other ones, not so important. You don't need to really know slide 3, slide 4, slide 5, etc. Slides 8 to 11 are important for you on your final exam. Slides 13 to 14 for RNA inheritance. You should know some of the methods. How do you actually determine if there are generational effects when you introduce an exogenous RNA? Okay, so you're looking at exogenous RNA, they eat bacteria, they look for GFP expression. Like, how do you actually look for generational effects? And these are outlined for you in slides 13 to 14. You don't have to know all the details of this paper. You should know it's C. elegans. So I told you at the very outset, C. elegans, no genes that they talk about in this paper. They do talk about genes in this paper. If you're totally lost about what that means, go back and revisit slides 13 to 14. It explains that. And then you should know some of the general principles. You are providing an exogenous or exo RNA that they eat that affects their endogenous RNA processing. There are two different pathways. You don't have to memorize all the details. The exo versus endo RNA processing, how that might lead to a generational effect. And this, again, gets back to this point about C. elegans and genes. There are genes that we talk about here, somatic expression genes versus germline expression genes. You should be clear on the difference. Do you guys know any without looking at your notes? Yes? Kai?
PEI is from the
Okay.
germline one.
Okay. Any other ones that you guys remember? Yep.
DPY2 is
Yep.
somatic.
DPY2, PPI1, I think, or PPI2. One of the other ones are germline versus somatic, and you should make sure that you're clear on those, okay? Because they're really important. So they wanted to know affecting the expression germline versus somatic. Okay, any other questions about this at all? Jeff, do you know if the recording kind of worked? Is it still going?
Yeah, it's still going.
Okay. So I'm going to try to make the link available for you, and then we have different audio recordings and the slides themselves. They'll be posted for you in case you came in a little bit late, or if there were things that you might have missed while you were trying to jot things down. Do you have any other specific questions before we wrap things up? Yeah, Kai?
Well, we didn't talk about IQ at all.
Nope, there's nothing on IQ.
So nothing on IQ?
Nope. I don't want you to waste time. I really don't. I think that IQ lecture was kind of interesting, but with less than half the class there, I didn't think it was going to be that fair to ask everyone. Or maybe it would have been fair, I'm not sure. Anything else that I can answer for you? So does this help you a little bit? I'm hoping to guide your sort of studies as you go on. Yeah, do you have a question?
In terms of like infectivity and transmissibility,
Yep.
Did you just mean like infectious doses and stuff like that, and then for transmissibility, like modes of transmission, like contact, aerosol, all that kind of stuff? What did you have in mind?
What did I have in mind? I guess there's a couple of different elements that I'd probably want to see. So one, do you have a clear idea about what transmissibility actually means? So here's a good example. I take, and this, I'm sure that this will be an answer on the final exam, even if I say it now. I'll take some of the misfolded protein and inject it into the brain of an animal model that, like a mouse, and then I'll wait for it to incubate. Then I'll go back and I'll see if there are any degenerative changes. So is that a good test that I just described for you? It's basic, but is it a good one? What would you mark that out of 5? Does it meet all the criteria that we talk about for prions? What's missing from that?
Detail.
Okay, definitely more detail. So for example, I would want to know if you have any idea that it's not just any old protein. If you're injecting in like 1 gram of protein into a 3-gram brain, sure, you're going to get all sorts of effects there. So talking a little bit about how much this is being infectious, like the relative size of the dosage. Is it like, really big, or is it really small that you start off with, and then how do you track it? So there are lots of details missing. So if I were to mark my own answer that I just gave you, I'd probably give it a 2.5 to 3 out of 5. There's lots of details that are missing, and they're in your notes, like the types of things that we'd want to follow through on.
So essentially just like a lot of detail and like infectious dose, 50 kind of like.
Those aren't the only criteria though. So if it stops at that 1 brain, it really doesn't tell you anything, right? And you need to be able to demonstrate transmissibility. Yeah, Oriana?
So just to make sure about the definitions, right? So for infectivity, is that more talking about like what kinds of cell types it affects and at what dosage? Whereas for transmissibility, that is, can it be transferred from the proteins of one animal?
I think what we're really talking about is transmissibility in this particular case. I'm not so concerned about the cell types that you might be affecting, like this area of the brain, if you want to, and the question asks for it, like it'll be really clear. But I think here in this context, we're talking more about transmissibility.
So you said infectivity and transmissibility?
Transmissibility.
You're
Okay. Anything else I can answer for your group? Ready to go? Shall we just write the exam now? We'll just do a special sitting just for you guys. Ready to go?
I just wrote an exam.
Sorry? No?
I just wrote an exam.
Oh, you just wrote an exam. Okay. So again, I'm around the next couple of days. So if you need to come in, there's last minute things. This always happens. Like you think you know it, you feel pretty confident, then you go back and review your notes, and then suddenly like, I don't think I understand it anymore. If I can help you out, let me know over the next couple of days. And otherwise, you can email me. There might be some really quick point that I can just clear up for you, or just to let you know if it's on the exam or not. And is that all clear on the recording side then, Jeff?
Yeah.
We're good to go? Okay, so I'm hoping it all worked out well. All right, it won't be terrible. It won't be easy, but it won't be like terrible. The only thing that you're going to be spending lots of time on is the writing part. So make sure that you're reading the question, underlining the things that you need to clearly have in your answer, and then make sure your answer is as complete as possible. Study the way that I've told you, the multiple choice, only 20 of them, things related to specificity, and for the short answer questions, make sure that you get all the points down that they're asking for. Develop your rationale clearly, 1 or 2 sentences, that's it. And then make sure that you've gone back over those 2 lectures that I've specifically highlighted for you toward the end. Okay, good luck. I'll see you all on the 20th. You have a last question. Yes.
Yeah, do we have like fill the blanks type of diagram? which you had
Nope.
in the
Nope. 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 Your bonus questions are there. Your bonus question is like a blank. Okay.